Hello children, welcome to biology class. Last class we discussed about replication. What is replication, children? Yes, it is the unwinding of DNA, isn't it? Children, it is now proven that DNA replicates semi-conservatively. Means half of the strands are conserved in the new strands. It's shown first in E. coli and in higher organisms such as plants and human cells. And two scientists, that is Matthew Michelson and Franklin Stahel, in 1958, they performed an experiment and they proved that DNA replicates semi-conservatively, means 50% of the strands are conserving during the production of new strands. Okay. So last day we discussed about replication. This is unwinding of strands. See children, here two strands are unwinding and new strands are forming, and during that time half of the strands are conserving. Is it clear? Now let's discuss the experiment of Stahel and Michelson, and this experiment is termed as Michelson-Stahel experiment. Okay, children, the first step, they grew E. coli in a medium containing NH4Cl. And what was the specialty of this ammonium chloride? The nitrogen, that means 15N, is a heavy isotope of nitrogen as the only nitrogen source for many generations. See? So for our understanding, it is given in brick red color and normal nitrogen 410 is given in an orange color. Okay children, so that's the first step. Then the result was that nitrogen 15 was incorporated into newly synthesized DNA as well as other nitrogen containing compounds. See, the first generation is having only N15, and in second generation, see, both N14 and N15 is present. This heavy DNA molecule could be distinguished from the normal DNA by centrifugation and cesium chloride CaCl density gradient. Children, this is an asked question, okay? How we can distinguish it from a normal DNA? By centrifuging it in cesium chloride. Children, please note, N15N is not a radioactive isotope and can be separated from 14N only based on densities. Okay. Then the second step, then they transferred the cells into a medium with normal NH4Cl, 14 NH4Cl, and took samples at various definite time intervals as the cells multiplied and extracted the DNA that remained as double-stranded helix. See children, this is the result after 20 minutes. Here also the color changes you can see. Okay. Then, the various samples were separated independently on cesium chloride gradient. For what? Yes, to measure the densities of DNA. And what was the result? The DNA that was extracted from culture one generation after the transfer from 15N to 14N medium, that is after every 20 minutes, okay, and had a hybrid or intermediate density. Children, why they took the time as 20 minutes? Yes, E. coli divides in every 20 minutes, that's why. Okay, and the DNA extracted from the culture after another generation, that means after 40 minutes, was composed of equal amounts of this hybrid DNA of light DNA. See, light DNA and hybrid DNA of equal amounts. Clear children? And this proves that the half of, that means the DNA is replicate semi-conservatively, means half of the DNA fragments, that segments, are conserving for next generation. Children, let me ask one question. If E. coli was allowed to grow for 80 minutes, okay, then what would be the proportions of light and hybrid densities of DNA molecule? Can you identify the answer? Can you say the answer? Yes, after 80 minutes there will be 4 generation, isn't it? In the first generation, all the strands will be hybrid, as the heavy isotope will be incorporated in the newly synthesized strand of DNA. That is, the new DNA strand will be of intermediate nature. In the second generation, 50% of the DNA will be light and 50% will be hybrid. It will be the result. After 80 minutes in fourth generation, it will be the result. Here also you can see the DNA replicate. You can say that the DNA replicate, yes, semi-conservatively, isn't it? And children, the very similar experiment is conducted by Taylor and colleagues in 1958 by using radioactive thymidine, okay, to detect distribution of this newly synthesized DNA, and they proved that experiment also proved that the DNA and chromosomes also replicate semi-conservatively. Clear? Now let's discuss the machinery and the enzymes for the process of replication, for unwinding. The DNA has to be unwound, isn't it? In living cells such as E. coli, the process of replication requires a set of catalyst, means enzyme. The main enzyme is DNA polymerase. Children, from that word itself, it is very clear that the reaction is polymerization, isn't it? Lots of nucleotides here, DNA is going to synthesize. That's why the polymerization of deoxynucleotides are going to happen, isn't it? So for that, for this polymerization reaction, for producing DNA templates, which enzyme is required? Yes, DNA polymerase is required for adding deoxynucleotides. Clear? Then, these enzymes are highly efficient enzymes as they have to catalyze polymerization of a large number of nucleotides in very short time. So, this is an example in E. coli that has only 4.6 into 10 raised to 6 base pairs. Compare with human, those diploid content is 6.6 into 10 raised to 9 base pairs. Okay, children. And it completes the process of replication within 38 minutes. Can you imagine how fast this process is happening? That means the average rate of polymerization has to be approximately 2000 base pairs per second. So, this much fast is the reaction. Okay. So, children, if any mistake during replication would result in a mutation. While discussing about the diseases, genetic diseases, we explained a point mutation, a single base pair change. It results sickle cell anemia. Do you remember it? Yes. So, furthermore, energetically, replication is very expensive process. Deoxyribonucleoside triphosphates serve dual purposes. Okay. In addition to acting as substrate, they provide energy too for this reaction. Okay. Energy phosphates, same as in the case of ATP. So, they are acting as substrate. At the same time, they provide energy for polymerization. In addition to DNA-dependent DNA polymerase, many additional enzymes are required to complete this process. Children, for long DNA molecules, since the two strands of DNA cannot be separated in its entire length. Why? Yes, due to very high energy requirement. Okay. So, in one strand, the complete synthesis polymerization is taking place. We can call it as leading strand. But here in another strand, it is not occur completely because of this energy requirement. Okay. So, the replication occurs with small opening of the DNA helix referred as replication fork. This is replication fork. A fork-shaped structure is formed. So, DNA-dependent DNA polymerase catalyze polymerization only in one direction. That direction is 5' to 3'. Can you see here? This creates some additional complications at the replication fork. So, see here you can see a continuous strand here, a discontinuous strand. This discontinuous strand is also known as lagging strand or Okazaki fragments. Children, why it is like this? Yes, because of this high energy requirement. So later ligase will join this strand. Okay. So consequently on one strand the replication is continuous, another strand is discontinuous. The discontinuous strand later joined by DNA ligase. The DNA polymerase on their own cannot initiate the process of replication. Isn't it? So also the replication does not initiate randomly. So here some regions are required. It is termed as ORI, origin of replication. Here the process of replication, that means the polymerization, addition of deoxyribonucleotide starts here. It is because of the requirement of the origin of replication that a piece of DNA, if needed to be propagated during rDNA procedures, requires a vector. Children, while explaining biotechnology, we had discussed this. Children, in eukaryotes, the replication of DNA takes place at S phase. One more question you can expect from here. The replication of DNA and cell division cycle should be highly coordinated, and a failure in cell division results to polyploidy. That means a chromosomal anomaly. Now let's discuss the difference between leading strand, is continuously formed strand. Here small fragments, Okazaki segments. We can call these small fragments are Okazaki fragments. Here it require only a one primer, but here small fragments are formed, isn't it? That's why separate RNA primers are required. Here no need of DNA ligase, no need of ligation, because it's a continuous strand. But here small fragments are formed. That's why DNA has to be joined. And direction 5' to 3'. Here complete strand is 3' to 5'. However, for Okazaki fragments it is 5' to 3'. Okay. So that is about the process replication. Clear, children? So you study this diagram. Study this diagram well. So here you can see at the origin of replication what? At the origin of replication, ORI, replication starts. RNA primer starts the process of replication, and at 5' to 3' in one strand the continuous polymerization reaction takes place. It is known as leading strand, whereas on other strand the continuous polymerization is not possible because of the energy requirement. So small fragments will form by adding a piece of RNA primer. Okay. Then later DNA ligase will ligate the strand and two new strands. That means the unwinding of strands happen. Now our DNA is unwinded. Then starts the next step, that is transcription. So now the unwinding is done. Then from this unwinding new RNA strands are going to synthesized. This process is known as transcription. Okay, so from DNA, RNA is formed. It is the only process of copying genetic information from one strand of DNA into RNA. In transcription, only one segment of DNA and only one strand is copied. And here, what is the difference between, an important difference between DNA and RNA? Yes, here the nitrogen bases are adenine, thymine, guanine, cytosine. But here in RNA, instead of uracil, yes, thymine is there. So transcription of DNA include a promoter, the structural gene, and a terminator. The strands that has polarity 3' to 5' act as template and called template strand, and other strand called coding strand. Okay, children? So let's discuss the process of transcription in detail.